Here today we have Zhongyu Yang um, as a guest for our IVM seminar series, the last of this season that concludes this first 6 months of the academic year. Let me tell you a little bit more about Zhongyu. He's from China. He graduated from the University of Science and Technology, and then he came to the US to do his PhD, and he worked in Pittsburgh with Sunny Saxena. And there, of course, he started working with EPR. He then moved to UCLA for a postdoc, and then he started his own group in North Dakota State University in 2015. He was assistant professor there, and he developed his research program which is covering kind of biochemistry, material research. I mean, it's pretty broad. And then very, very recently, like a month ago, he moved to the University of Missouri in the Department of Biochemical Engineers, something like this. And today is gonna tell us more about protein under confinement. So Zhongyu
Okay.
can
All right.
start.
Thank you, Thomas. And I also want to thank the IVEM committee. It's been a couple of years since I heavily involved in IVEM activities. I think I officially stopped when I get my third SFPI grant. I was just super, super busy and I couldn't deliver what I committed to. And that's 2022, mid-late 22. However, I'm glad to see how the IVEM develops, the audience developed, the committee member all grow in their independent career. I mean, that's the whole spirit, right? It's not for any single person to hold this committee forever, right? It's an activity that helps our whole EPR community. Now, as Thomas said, I was in North Dakota, Fargo for 8.5 years, almost 9 years. And then I started with a biochemistry slash bioanalytical division at NDSU, and my teaching was all in analytical chemistry. So initially, I just want to do protein dynamics, right? I study the challenging protein, solve some biochemical problem. And then over time, I realized that something else that interested me more. So, namely, the interface of proteins and the nanoscale materials. Initially, of course, we start with nanoparticles. It's on the surface, so we call it a positive curvature. And recently, we are interested in negative curvatures. For example, compartments, right? So where's my laser pointer? Does it work?
Yes,
So
yes, we can see it. Yeah,
this one?
yeah, it's
Yeah.
all good.
Oh no, hey, where's the laser? Yeah, I'll just use my mouse. So and then later we become interested in negative curvature, meaning confine proteins into synthetic compartment materials. And then, therefore, over the years, we begin to study different proteins, study different porous materials, starting right mesopore silicon nanoparticle, uh, carbon nanotubes, and all the way through metal organic frameworks, covalent organic frameworks. What we are more interested in is the how to say the synergy or how the synthetic and the natural materials how they interact with each other to generate new functions, new materials, or deliver us new knowledge. Right. So this is the basic journey that I'm talking about, and like I said, I just started lab like 4 weeks ago, and then. Therefore, there will not be too many new data that you're going to see. So let's see. But we will be doing the same research, also confining functional biomarker molecules into synthetic compartment materials at Missouri. So when we talk about materials, I kind of want to give a general summary. So what materials we're talking about, right? This include sustainable materials where we actually confine specific enzymes that can degrade the plant biomass, right? Different components of plant biomass. So that will help break down plant cell walls and release the valuable components, valuable species inside, right? And that's we call it sustainable materials. Energy materials, we can confine a few redox enzymes, FDH, right, and ADH. They will stepwise convert carbon dioxide into fuels. Biomaterials or biomedical materials, we confine proteins, especially interested in HIV proteins, to degrade different proteins, polypeptides, and also to test different protease inhibitors to help drug development. We also cover the polymeric materials, basically polymeric self-assemblies where you can load polypeptide proteins and the DNAs, right, into the assembly and help the delivery. And we put a lot of efforts on understanding the dynamics of the delivery. We also use we want to use the confinement compartment materials to help biophysics, right? To help biophysics, basically we can use systematically synthetic materials with systematically alter the surface property and look at how protein structure dynamics and function changes in there. And that's for fundamental biophysics. We also tried to understand how disordered proteins are translocated into mitochondria through some protein conducting channel. So yeah, there are a lot of projects going on in my previous lab. I mean, unfortunately, my current lab, we can only do a few of them. But many of this funding from NSF and USDA, as you can tell, many of this funding are still active. Even my earliest career award, I just get no cost extension. So that's a basic overview. How can I see? Yeah, since this is a EPR meeting, and then I want to focus a little more on the EPR and the biophysics side. So the key questions we want to ask in this many materials that I just listed for you, what are the structural dynamics of functional biomarker molecules, especially enzymes, when you immobilize them in these materials, and how do they interact with a solid surface, solid matrix? What are the orientations of enzymes in this solid matrix? How strong are the interaction, right? Are there any aggregation during immobilization, especially when right, you're dealing with polymeric materials, right? And then lastly, how do proteins translocated into this space and fold they're in, right? Now, all of those are not super challenging or super difficult if you just read from the word, right? By word, it's not that hard. However, it is the compartment materials that make it more challenging, right? The key barriers is, of course, the shielding effect of this compartment, right? Either polymer or MOFs, or and also the how to obtain sufficient time and spatial resolution when you confine these proteins into those materials. Those are the two key barriers. So, however, I mean, those are the questions my group is trying to answer, to address, right? So, in fact, chasing the answers to these questions, that's what makes my group different from a regular classic materials research group, right? Because we care not only about the right, the function, right, the novel applications, but also we want to understand why the enzymes behave specifically in different materials. Now, to overcome these barriers, we have the near perfect tool, right? That's the spin EPR and the spin labeling. So the direct spin labeling, I guess I don't need to introduce too much to this audience, right? Make a system, put a MTSSL, generate R1 spin label. That's how both work, right? And it's still very solid, very applicable nowadays. And once a protein is labeled as at a specific residue, right? There are three motions mainly, right? Protein tumbling, helix rocking motion, and intrinsic motion of the label, right? They all contribute to the EPR spectrum. And by EPR, I'm talking about a CWEPR only. Right. So reality is I only have CWEPR spectrometers in my lab, and I want to see how to take the best use of it. And it turns out to be very useful, actually. So you all know this, right? Rate of motion, right? As the motion goes faster, right? The line shape becomes sharper, right? The three lines becomes nitroxide. And you also know this order of motion, right? As the motion becomes more ordered, meaning as the space allowed for the spin label to move around is more restricted, right? The line shape also becomes broader. And we have the famous MOMD model developed by Jack Fried's group that can simulate the spectrum. And when you have multi-component, meaning with some protein right, interact with something with the label site, but the rest of protein does not, then you can deconvolute through spectral simulation too. So I'm going to show a couple of applications due to the matter of time today. So first is we want to understand how enzymes are immobilized or crystallized in meta-organic frameworks, right? Or during the crystallization of MOBs. So the earliest example we have was zinc imidazole and enzyme. You put a PVP polymer to protect the enzyme, put everything into methanol, and then make this we call it a co-crystal or biocomposite or whatever you want to call it. So no enzyme with enzyme, you can see there's some bumpy surface. And then so here is just the structure of the MOF itself. So we characterize the MOF with powder XRD, TGA, and nitrogen isothermal, right? That's a porosity test. All confirm the presence of enzyme, and then also the enzyme did not significantly interrupt the crystallinity. And then our question is, what happened to this bumpy surface, right? Are the proteins just there to mess up the crystallization, or are there any other reasons? So we later did some activity tests, and here's our hypothesis. The surface is bumpy because some proteins are partially trapped on the surface. And TEM, this is a TEM image, right? TEM does not see proteins, unfortunately. So our hypothesis is that these enzymes are partially trapped, partially protected, but also partially exposed. And therefore, you can do activity tests, especially when the substrate of this enzyme is huge, right? We're talking about a micrometer substrate which cannot diffuse into this pores of the mouth, right? So exposure of active site of the enzyme above the mouth surface, it becomes important. Now, if this hypothesis is correct, right, then we have a way to immobilize enzyme with no enzyme and substrate limitations as limitation, and we offer partial protection to the enzyme, which later in our later works proved to be very handy, very solid, right? So to of course, to prove this hypothesis, we immediately try a lysozyme enzyme which degrade bacterial cell walls, and cell walls are micrometer order, which cannot diffuse into these small pores. So we see activity that proves the enzyme is partially exposed. We wash the co-crystals, and then the activity is still there. That means it's not because of the enzyme physically attached to the surface, right? It got to be right. It got to be geometrically trapped on the surface. So you wash it, you don't lose the activity. And the next question is what's the orientation, right? Are all the enzymes oriented the same way, right? The same orientation, or they are random? Now, to understand or to review the exposure area and orientation on the surface, we use the spin labeling and the EPR. And the idea is quite simple. I mean, if a labeled site is buried, you see highly restricted motion, right? We call the immobile peak. If the labeled area is exposed, then we see a mobile peak, right? Meaning they have a very different, a very distinctive rate and order in their motion, and we prove that in simulations. We also can use a interesting finding that urea does not unfold the proteins buried inside the mouth, but only unfold the proteins exposed. And we do the same thing, right? The immobile peak does not change. The exposed area component get sharp. That's because of unfolding. And we do the same thing on multiple surface site of the enzyme. And then basically there are a lot of data, the same idea. Find the two components, use urea to unfold the mobile component, and the immobile does not change. We also run simulations. We understand the relative contribution, the percentage of these two components. Now, there is for one side, we have about 33 plus minus 2% of exposure. That means right about 1/3 of the 44 site, which is here, is exposed. But through the whole protein, right, in all these sites, multiple sites they show similar percentage. There are other sites they show much less percentage of exposure. And that means, right, orientation cannot be uniform, right? It got to be random. Meaning there's no site that's completely buried, 100% buried in a way. So the orientation is more like random. And we quantify through simulation, we quantify the exposure percentage of every residue, and we found that the first orientation where we expose the N-terminus, right, especially the blue star that's the active site, right? This orientation is the most populated, it's about 33%. And the second orientation is less, and the third orientation that exposes the C terminus and bury the active site, that's the least possible orientation. That's about 10-12% of chance. Now, we also try to understand what has been buried using a different morph. So, I mean, essentially they're the same zinc. It's called the zinc imidazole morph, right? But this time we make the ZIF not in methanol, but in water phase, where we lose crystallinity, and we see more heterogeneity in the local backbone motion restriction through many of these sites. And we kind of not only in this nanoscale work, nanoscale 2023 work, we not only reviewed a similar exposure area, but we also tell the differences inside of the morph. Now, this work is quite interesting because it indicates that using EPR you can, right, not only tell the orientation exposure, but also you can tell what's the crystallinity, right? How well the crystals are packed inside. Because clearly here there are some residues that show more activity, some are more restricted, etc. So we therefore want to try. I mean, I'm still a EPR guy. I want to see what the EPR can do, right? So my question to my student postdoc now is that is there any way we can use the EPR to prove, right, how EPR can tell the packing quality, the crystallinity, right? But to do that, you need different morphs, right? You need a mouse with different personality. So we did a lot of trial and error. We found five cations and two ligands, right? It's just a benzene ring with two carboxyl, two benzene ring with two carboxyl. We call that BDC, call that BPDC. So 10 morphs basically. And we found that some of the morphs, they can form crystals. Some are multi-phase crystals, and some are completely amorphous. And that's perfect for my goal, right? So we load different a couple of enzymes and we check the reusability. The both enzymes on most of the 10 morphs are highly reusable. So which is good. That means the enzymes are trapped inside. And crystallization was not significantly changed upon enzyme binding. And some uh, crystals like zinc BDC, right? Those are zinc BPDC. Those are kind of almost near single crystals, very crystalline or very, very high crystallinity. And some we have to feed the powder XRD data with a multi-phase. And some we cannot feed anything like the current BDC, the current BPDC is completely not crystal. But still, it's a solid, it feels like a gel. So now the EPR comes to place. So I told you how we can distinguish the exposed and buried site. And in fact, if we look at the mobile component, it not only tell the local motion of the labeled site, but also tell you how the packing quality at the crystal surface. And inside, right, the immobile peak, right, not only tell how restricted the label site is, but also tell you the packing quality inside of the mouth. And of course, the relative population tell you how much exposed. So by doing that, we found I know I show you 10 mouths, but there's a copper BPDC, copper BDC mouth. The copper signal mess up the nitroxide signal. So we're still analyzing those data. But with these 8 mouths, we already found that as we go, I mean, if you remember the previous slide, right, zinc BP, right, this is higher crystallization and this side is lower crystallization. So as we decrease in crystallization, the decrease in crystallization, the R value is a kind, the immobile component of the R value is a kind of decreased. Meaning, right, the lower the rate, meaning the slower the motion, right? And then, so this is what we propose, a single crystal sharp, right, multi-phase, right, that's the real data. At the very end, you can see there is a jump, right, in the, especially the BPDC, there is a jump in the motion, in the R rate constant, right, and that is because when it's not crystal, and they're still solid, it feels like a gel. So when there's a gel, you have the highest rate. Now, comparing BDC to BPDC, you can see even though the length of BPDC is longer than BDC, we actually see an increase in crystallization, therefore reduce in rate. That means right, even though the linker is longer, you can form better crystals. I said except for the zirconium, the gel, that's because when you form a gel, right, the longer the linker, the bigger the gap, and the more the higher the rate, right? So basically, we use the EPR fitting parameter. We figure out a rough way to correlate the crystal packing quality with the rate parameter of EPR. We, of course, are not so happy with the quality. That's because let me go back, all of these morphs are made in water phase. And you know, when crystals are formed in water phase as compared to organic phase, you have a low crystallization and low yield. So we introduce the liquid system grinding method. So basically, you have a tube and you have a stainless steel beads, and then you rotate the tube so the beads are enhancing the interaction between the enzyme and the liquid, right? And the setup is quite simple. Everything is room temperature. It's aqueous, but we put some salt, and then we did see a significance improve in some of the multi-phase or amorphous morph. You can tell in the black data, there's a more sharper peaks, and all of the all of the enzymes are active in this liquid assistant grinding, like synthesize. stuff. We're running simulations, and hopefully we can have that paper out. So, uh, so this is like the first application. We use co-crystallization to trap enzymes during the morph formation. The second application I want to show you is all about unpublished data. So the idea is that we want to understand the protein folding in a real cellular environment. That's because there are many spatially confined cellular compartments, right? And proteins, even though our protein folding studies are mainly in dilute solutions, right? In cells, proteins are folded under confined space. So it's difficult to study protein folding in real cell, right? That's because of the complexity. I mean, you can do NMR, you can do an in-cell NMR EPR, but it's quite complicated. The other alternative is to mimic the cellular environment. And Martin Krubel and his co-workers summarized that the cellular environment basically can be separate disentangled into crowding effect, right? Sticky interactions and the confinement effect. Now, the first two, those are relatively straightforward to mimic, right? Just mix in different crowders or change the solvent, then you see it. The challenge, however, it is the confinement, right? That's because it's hard to control and mimic the size, shape, hydrophobicity of the cellular compartment. So what we do, right? We mimic again, and one option is native chaperones. Those are good, right? Mimic the complexity and the dynamics. However, it doesn't offer too much chamber, right? There are only limited chaperones in nature. And it's hard to study protein folding in chaperones unless your protein is already disordered, loaded in, right? Otherwise, you can unfold the protein, you load in, you want to control the different, say, urea concentration, but in the same time, the chaperones will be unfolded. Now, the other option is, of course, synthetic compartments. They're easy to control, stable in under denaturing conditions. However, we don't know how well they mimic the cellular conditions, right? So clearly, some comparisons, some extensive work is needed. So what we want to do is to start with the synthetic materials because we have them. And of course, I'm trying to work with collaborators on native chaperones too, and eventually make a comparison and see which morphs can mimic the chaperones better, right? So our first attempt is quite preliminary. So we try to, I mean, as you can imagine, right, if you want to study protein folding using EPR, right, you control the different urea concentration, right, you can study both the unfolding and refolding process, etc. So lots of data needed. So we want to limit ourselves. So we narrow down to 4 synthetic materials, right? There's one COF, that's PCN333, that's a natural cage. PCN128, it's also neutral, but it's in a channel shape, it's long, right? This is a cage shape. Covalent organic framework 2, we call the COF2, that is a hydrophobic channel with a similar diameter, but very hydrophobic. A narrower hydrophobic channel called OME, right? You can see the difference, right? There's about a 1 nanometer difference in the channel. So channel diameter. So by comparing like each two pairs, right, we will be able to tell say comparing these two, we can tell roughly what does size do to the protein folding. Compare these two, we can know what does the hydrophobicity do to folding, right? Compare these two, we can review the effect of shape, right? So we use the same model protein. We label 6 or 8 sites on the surface. And then our typical data look like this. We study both the folding process. By folding process, we start with loading the protein with under 8 molar urea, and then slowly, stepwisely reduce the urea concentration to 0. We call it folding, right? And the black is unfolding, so we go from 0 native protein load into each of these chambers. We gradually increase urea concentration until 8 molar, right? And we run simulations. We plot the folded percentage as a function of urea. So there are a lot of data, a lot of simulations, as you can tell, but to just give you a summary of what we find, right? So comparing solution versus confinement, right? Buffer versus COV2, you can tell the activity completely lost, but COV2 kind of prevent the complete loss. There's still some remaining 40% or so, right? And then the CWEPR in solution kind of reflect the similar complete unfolding, and refolding is kind of a reverse. But in COV2, you can tell there are significant amount of 44 sites and 151 sites, which are representative of the N and C terminus. They do not completely refold and do not completely unfold, and we want to do refolding, they kind of go back. So this tells that the confinement does something, right? Confinement prevent the complete unfolding, complete loss in activity, right? Now, when we want to understand confinement size on protein folding, we compare the data, right? COV2 was the same data before, but this time it's COV-OME. You can see the activity loss in COV-OME is even less, meaning the smaller chamber kind of prevent the complete unfolding, right? Try to protect the protein from unfolding. I mean, that makes sense, right? You want to unfold the protein, you need sufficient entropy, you need enough space, right? And EPR also tell you similar story here, except that the unfolding and the refolding are not reversible, right? That tells us, probably indicate that there's some heterogeneity in the unfolding and the refolding process, right? Maybe some hidden folding intermediate or hidden pathway. And to look at the impact of hydrophobicity, right? Comparing COV2 with PCN128, that's the same data, right? The difference here, similar size, the same shape, but different, right? This is neutral, that's hydrophobic here. So you can see neutral channel does not protect that well, and in neutral channel, right? Especially refolding data, right? You lost more, and those are kind of consistent. You can see orange consistent with orange. So hydrophilic channel kind of right does not help prevent the unfolding of the protein. And now channel confinement shape, right? The shape is compared between the channel, right, PCN128 and the cage shape. You can see it's even worse, right? In cage PCN333, we lost a lot. Look at the EPR and the activity, right? We both lost a lot of secondary structure, meaning somehow the cage shape PCN333 does not protect the protein unfolding as much as the channel shape. I mean, it's probably also because this protein is like an oval shape, right? Rectangular shape, so the channel favors that. I wonder if we switch it to a spherical shape protein, is the same conclusion. So excuse me, my room is cold. So we study both unfolding, refolding, right? Solution is reversible, but when under confinement, right, some are reversible, some are heterogeneous, and different confinement factors, right, shape, size, hydrophobicity, they have different impact on protein folding. So overall, we just use CWEPR, if you want to start independent career, right, and if your school cannot afford a pulse EPR immediately, right, CWEPR is good enough to tell us a lot of information. And I'm only showing a small piece of my data. I mean, we probe the exchange interaction, we probe the aggregation, right? We do time resolved CWEPR to study. So the take-home message is CWEPR is also quite helpful. Now the people I have I had a group of 6 or 7 people, and somebody borrowed borrowed by 2 groups. Currently, I only have 2 graduate students and 1 postdoc in uh, Missouri. The work has been supported by various funding agencies. And then the spectrometers are donated by EPR pioneers Peter Fayer and Stephen Chen. Right, Peter Fayer retired a few years ago from Florida State University. Stephen Chen was a NIH investigator, later moved to NDSU. He's a cancer radical EPR guy. Maybe some of you already know that. He passed away 6 years, 5.5 years ago. But he donated his spectrometer to my lab. So I'm really grateful for these pioneers. So with that, I will stop and then answer any questions you have. Oh,
Thank
did I
you. Thank you,
stop
Zhongyu,
sharing? Yeah.
for your seminar. That was very, very interesting. Of course, the audience everyone is encouraged to ask question. You can, I mean, we are a small crowd, so you can unmute yourself and just ask question to Zhongyu. I would start with just a couple of very trivial ones. So, so what is the size when you study the protein in the mouth in the second part of your presentation? I missed what is the size of the protein? You said it has an oval shape. Uh, do you know what's the size in the 2 dimension with respect to the channel?
Oh, so here that's probably so usually the protein is like 2.5 by 3 by 4.5 nanometer 3 dimension.
Okay.
And then the channel is usually,
Oh,
I mean, we control that. So this
yeah,
one is
exactly.
4.5, that's 3.4 nanometer. And the cage is 4 point something nanometer size.
So the channel and the size of the proteins are comparable.
Yeah, they are actually quite close. I mean,
Yeah.
let me keep sharing the screen. Yeah, they are actually quite close, as you can tell. This
This is to scale. The drawing is to
Yeah,
scale.
almost
Almost
to
to
the
scale.
scale. Yeah,
Okay.
almost.
And I have another question, like, does the change in activity, the change in activity depends, of course, on the urea concentration. Um,
Yes.
what is the concentration of the protein? Does it depend also on that? Maybe when you pack more protein, you kind of prevent the folding, or does mix?
Okay. Good, yeah, good question. So we we struggled a lot try to try to normalize the activity, right? Because depends on how much loaded and then also restriction, right? Substrate diffusion, etc. So for this activity test, it's the same enzyme, but this one we cannot use a bacterial cell wall as the substrate. We use a smaller substrate that can diffuse into the channels and the cages of the moth. So diffusion rate, right, loading capacity, that is very difficult to calibrate. So loading capacity we can do because we have a BCA test. We can also do before and after. So we put extra protein inside, or we wash, and then we
Ah,
see how much is removed. Those we can quantify. So loading capacity, we have a pretty good idea, and we kind of normalize the activity according to the loading capacity. However, we really don't know the diffusion rate, the diffusivity of the small substrate, right? Because the substrate had to diffuse in, get cut, and then release, so we can measure the activity. That we have to assume because the substrate is quite small, it's like less than 1 nanometer, and we assume they can go in and out. But we're looking for theoretical measurement or NMR measurement of diffusivity of that substrate through this MOFs. So we I'm sure we must correct the diffusivity when we discuss the activity. However, the activity trend seems to be close to EPR trend. So
Right.
yeah, I mean, you understand it's a very complicated system. It's a mess, and we need to trim out key information out of it. Yeah, but it's a good idea. We need to think about it.
Great. Thanks. Are there any questions from others? If not, I have one more actually. How fast is so what is the time scale here of the single EPR measurement and also of the whole process, like folding and unfolding? Like how long does it take? And
Yeah, so good question. So for example, if I want to say fold the protein, right? I unfold it, use 8 molar urea, load it to the mouth, which was also incubated with 8 molar urea, right? So it's completely unfold even inside, and I measure EPR. And those I can do 5-10 scans, like 3-5 minutes. And then to trigger the folding, right? I wash the mouth or cough loaded with 8 molar urea unfolded protein with say 4 molar of urea. I wash a few times. I let it equilibrate for 30 minutes to 1 hour, right? And I wash again, make sure everything is a 4 molar urea, and then I run EPR again. So it is a thermodynamically equilibrated state, which we can run EPR for a few minutes. But EPR can do time resolved measurement, right? So I have no doubt that I actually have a stop flow system. If I do a stop flow and see how the signal change, that will be interesting too. So because with the confinement, I would say the kinetics is going to be slower. I would say as compared to because in liquid, in buffer, it's almost immediate. You drop some urea, the protein immediately unfolded. But urea need time to diffuse into the mouth, and then the mouth, the protein inside the mouth need time, the water, the entropy of water. So actually, some DNT measurement may be nice too, if you guys have the equipment.
yeah.
Right. So cool. Yeah, it's a good question. Good thinking.
Okay. Yeah, it's very interesting work. It's definitely something
Yeah, I
I'm
mean the
interested.
video. Yeah, you share the video online. So if anyone has questions, just email me. It's a
Absolutely, yes. You
Yeah.
can.
Just
They can.
Google my name with Missouri. You should find my I don't
Yeah.
know. You should find my homepage.
Yes. Absolutely. Yes. It's easy to find you online.
Yeah.
All right. Any questions from like last minute, last seconds? If not, well, thanks. We thank Zhongyu again for his nice seminar. It will be online. And yeah, happy Chinese New Year, by the way.
Oh yeah. It's a yesterday, but
Oh, it was yesterday. Okay, I thought it was
yeah, it's
okay.
officially yesterday. But
Okay,
I have
my
three
bad.
back-to-back Zoom meetings, so I just give myself a day off.
Okay.
Yeah.
Yeah, maybe the
Okay.
weekend you can.
All right. Yeah. Good to see you, Thomas.
Yeah, good to see you too. And
Hopefully
yeah,
this time is good because I can participate more. Previously, it was not good. So
okay,
yeah, hopefully I
okay.
can
Now that
participate
you're in
more at
Central
the
Time.
end. Yeah.
Okay, good, good,
Okay.
good to
Okay.
know.
I'm
All
leaving.
right, thank you,
All
everyone.
right. Have a good day, everybody.
Bye.
Bye.